Swayam Prabha. Digital India. Educated India.
Okay, so let us now get back to developmental biology. Okay, so where did we leave last? Differential gene expression. Okay, so with that we ended there. So we will continue. So before that we were discussing about early embryonic development, right? Pre-formation, epigenesis, those kind of things. So the next thing we are going to move into is morphogenesis. Okay. And so you will realize although in today's lecture we are primarily going to focus on discussing cell adhesion and cell shape changes and differences in their affinity for which cell will attach to which cell and those kind of issues. But the underlying thing in all of this is differential gene expression. Okay, so that is the core of all the changes. During embryonic development, the morphogenesis related questions, remember morphogenesis itself was one of the major questions we started with in the introductory class. So today we are going to look at some of the sub questions within it. So one of them is, like, for example, it is not in any particular order, but it is sort of developmental chronological order. These questions are not based on importance, all of them are equally important. So how are tissues formed from populations of cells? Okay, so you have populations of cells, but then each cell type has to organize into a particular tissue type, because many organs are made up of multiple tissues, right? The example used in the book is the eye development. In the eye, if you look at you have retina, and then you have cornea, lens, and so on, and within all of them together is an organ, the organ is eye. Okay. So now each one of these things, like for example retina is a tissue, and that is not made up of only one kind of cell, it is not one single set of cells that make that tissue. So a tissue may have multiple cell types. For example, here you have pigmented retinal cells and neural retinal cells, and sometimes you can have multiple kinds of cells in one tissue. So you need to understand this hierarchy very well. Cells, of course, you understand, different kinds of cells you understand, but what you need to understand is a given tissue might have multiple kinds of cells or maybe only one kind of cell, depends. And a tissue by itself does not make an organ, or in other words, an organ may not be made up of a single type of tissue. So it may have multiple tissues. That is where the difference between a tissue and an organ comes. Then you have to have organs in the right parts of the body. Okay, and in an organ also the tissues have to be organized correctly. So these questions concern about these aspects, like how the tissue forms correctly and how are organs constructed from tissues. The right tissues have to be at the right place to make the organ. But retina has to be at a certain place with reference to cornea, for example, okay, to make a functional eye. Otherwise you would not make the functional eye. You could have the right tissue, but then if they are not at the right places, then it will not form a functional organ. So how does that happen? And then the organs have to be at the right locations. Okay, no point in a particular cell type differentiating into cornea and retina in your spinal cord somewhere. Okay, it should form at the face at the right place, and it should have the right neuronal connections to the brain, the right part of the brain. So how do they all get to the right places and form those structures? And these processes involve cell migration as well. So how do cells migrate correctly? There are some tissues formed at the right place with no big migration, but there are certain other cell types that migrate a lot, like blood cells, for example, germ cells. They have the somatic gonad forms elsewhere compared to where the primordial germ cells are formed during embryonic development, and they have to migrate to the right place. So, how they migrate to the right place, and once they know the destination is reached and they stop migrating any further. So, how that is controlled. Then once, let us say, right cell types form the right tissues, and right tissues at the right location form the organ, and organ is at the right place, but it still needs to grow, right. Your eye is not the same as how it looked when you just came out, right. You look very different now. So, how they all grow proportionally, that is very, very important. So, how does that happen? And organs do have polarity, right, like from a limb bird your hand does not grow and fingers are formed anywhere in the hand, it has to be at one end. So, there is a polarity to the structure, right, every organ pretty much has polarity to it. How that is achieved? So, these are all the questions that come under morphogenesis. So, we are going to get a glimpse of this, we are not going to get detailed answer for all of this, then there will be no research left for developmental biology. So, we are going to get an idea of how these processes happen. So, that is today's topic. So, like any other topic, we begin with historical beginnings, how people started looking at it. So, this is one of the classical experiments where we very fortunately the frog embryonic stages, the blastomeres, the cells in the early embryo can readily dissociate if you put in an alkaline buffer. So, these scientists took advantage of that, they isolated cells in that way. Like for example, you take two different pigment colored frog embryos, and then here in one of them you are taking so from this one you are taking epidermal cells, and from here neural plate cells, and then you heap them together, okay, and they just do not stay like that. So, they reorganize, they seem to know where to get, and the dark blue knows that I need to get with all other dark blue cells and get away from this light green one, and they separate, and they separate in this fashion spontaneously all by themselves. Okay, cells do sort. So, that is the main observation from this experiment. Then when they tried with the different, so this is like the very first early experiment, now you try with the different germ layers, okay, and then see what happens. So, this is summary of those experiments. So, the summary here is the second point, that first one is the cells do sort, they do not remain randomly. Final position after sorting reflect embryonic positions, okay, in the natural embryo where a particular kind of cells will go, and that is where they go even in this in vitro condition when they are allowing them to sort themselves. For example, when you take epidermal cells, the blue colored ones, mesoderm, the red colored one, and then mix them, and then wait for some time, then they migrate, and then epidermis is outside, mesoderm is inside. So, that is how it is in an embryo too, the epidermis will be, the ectoderm will be outside, mesoderm will be inside. And then you do the same experiment with mesoderm and endoderm, and here you find they separate, but endoderm is outside, mesoderm goes inside when you have these two alone, which is not the embryonic localization, but when you mix all three, then you get the same as what happens in embryo. So, endoderm, mesoderm, and then you have epidermis. So, they sort that way. So, more examples here, neural plate versus epidermis, and little later when within a single germ layer, like for example in the ectoderm you got neural cells and then the epidermal cells. Remember, the pigmented skin surface cells come from the same progenitor as the neurons, right. So, they again separate, and when you have more varieties and mix them, again they separate the way they would normally do in an embryo. So, this suggested to Haltfretter that there is selective affinity. So, each cell seems to know with what it wants to interact and with what it wants to avoid interacting. Like for example, the mesoderm and epidermis, they seem to interact, and epidermis seem to always avoid the endoderm, and so on. So, there he proposed this idea that cells have selective affinity. Okay. So, now we are going to look at what is the basis of selective affinity. How do they determine that I have to be outside and the other cell has to be inside? So, that we get into another concept, but before that, this is not true just in the embryo, it is also true in adult body as well. Like for example, at that time alkaline solution was easy and they could isolate the frog embryonic cells, but now we know how to separate cells even from adult tissues, for example trypsinization and so on. And when you take skin and separate the cells into a pool of cells and allow them to organize, and then they finally organize back into the original organization, epidermis on the top, dermis below and hair follicles within that. So, they organize themselves. So, the next idea is how do the cells know where to go, and for that Malcolm Steinberg proposed a theory called differential addition hypothesis. Okay, so, they are still working on it, like last year when I saw they had a review recently. See, this started in 1960s, 1964 I think he proposed this differential addition hypothesis. So, it is quite simple. So, the proposal here is that the surface tension of different kinds of cells differ, and when they are mixed together they reorganize themselves such that the overall surface energy is minimized. Okay. So, it is thermodynamically more stable structure. So, that is what they are getting to. So, the embryonic arrangement is basically an equilibrium of these forces. So, the cells are organized such that the final structure is thermodynamically more stable. So, cells having higher surface tension or force of attraction among themselves, they go to the central part of the embryo. So, that is all is differential addition hypothesis. So, like for example, here the neural and pigmental retinal cells. See, retina is a tissue having two types of cells. Remember our very first point we discussed in the first slide. So, when you dissociate and mix them, then you find that whatever Haltfelter observed the same thing. So, here you have the pigmented cells go inside and the neural cells stay outside, and people in his group tried to measure the surface tension of individual cells, and they found proof for his original proposal, and that is what is shown in this. So, here you have the two kinds of cells that are being mixed in two different colors, like here green and red, and here red and yellow and blue, and then blue and orange and so on. So, in each pair if you see, the green has higher surface tension than this red one. Okay. So, the green goes inside. In the next between this red and this yellow has the lower surface tension, so as a result that goes outside, and so on to the next two pairs as well. So, cells sought to form aggregates with the smallest interfacial free energy. So, this is one of his students 40, he measured and showed this is what happens. So, the next is what is the molecular basis of this, how the surface tensions change. So, you need to remember like yesterday looking at the textbook you felt that the biochemical pathway is known, what is the big deal, but you needed to know the pathway, right, you need to discover. So, similarly when you do not know what is on the cell surface and there is something on the cell surface, and you need to know whatever is there that changes from cell type to another cell type. So, those information we need to know. So, initially people did not know that, they thought cell membrane is simply a lipid bilayer and therefore all cell surfaces were the same, right. So, these disciplines are simultaneously existing and developing together. Okay, a developmental biologist, or rather historically embryologist, is not waiting for the biochemist to purify and show integral membrane proteins, peripheral membrane protein and so on. Okay, so they are not waiting to know glycosylations exist and they are markers for like zip codes for individual cells. So, they are not waiting for that, they are parallelly studying. Similarly, genetics goes parallel, and developmental biologists are not waiting for the development geneticists to discover all the genes and show differential gene expression. So, you need to see in that historical context. So, that is how the ideas shape. So, here this was like a big progress. Right now for you it might look simple, yeah, there may be molecules that are different, but initially people did not even know there are things that are there on the cell membrane, and cell membrane of one cell could be different from the other. So, now we are going to look at the molecular basis for that. So, the molecular basis for this is rather extremely simple, it is not complex. These numbers are not brought by using the entire repertoire of our genome. So, only one group of molecules seems to be sufficient for this, and they are the cadagrins, okay, calcium dependent cell adhesion. So, that is what this word stands for, calcium, Ca, adherence, that is where the D stands for the dependence, and then adherence is adhesion. So, it is basically a calcium dependent adhesion mediating molecule. So, these are the really important molecules for cell adhesion. For I think most of the rest of the class we are going to talk only about cadagrins, okay, different experiments involving cadagrins. So, this is how a cadagrin structure looks like. So, they are embedded in membrane, so they are transmembrane proteins. Outside you have multiple distinct domains that bind calcium. So, these are the calcium binding domains. So, in this particular one you have 5 domains, and the very end of it has the perfect shape for interacting with another cadagrin molecule from an adjacent cell, for example, okay, as shown in the next one, the lower picture, okay. And if you look into the cell, like inside the plasma membrane, right side here, so you have sites where they can be phosphorylated, so that is one way of regulation, and then you have these catenins binding there, and what is not shown is the involvement of small GTPase called rho GTPase, that we will learn as we go through. Then these connect to actin cytoskeleton, which is on the periphery, inner periphery of cells, okay. So, this is how the internal cytoskeleton is connected to the external a molecule that goes and interacts with another cell. Of course, these molecules via integrin are going to interact with ECM also, but that is not our focus right now. Our focus is that this cadagrin molecules of adjacent cells interact, and that is how the cells are kept together, that is one. Second, these cadagrins are not simply anchored on the membrane, they extend into the cytoplasm where they interact with the actin cytoskeleton itself, and these can be regulated by these small molecules there. For example, this phosphorylation can cause this to dissociate from the beta catenin, or the catenins may be brought here or removed from, and actin polymerization, depolymerization also can affect this, and therefore, changes that happen inside can affect the external adhesion and opposite as well, okay. So, those are going to become clearer as we go through the rest of the slides. So, okay, so I said cadagrins are important. How do we know that cadagrins are important, right? So, this is an experiment for that. So, this is a frog embryo where you have antisense RNA, remember antisense we have already learnt, right, injected against cadagrin, and when you open the animal cap, these cells fall apart loosely, but in the wild type they remain together, they do not fall apart, indicating that the cells adhere normally, and that adherence is gone when you have stopped producing cadagrin proteins, clear. So, this is one experiment to show the importance of cadagreens during embryonic development. And if they are so important, then we are going to have variations in that theme, right. So, we have multiple types of cadagreens, like embryonic cadagreen, which is slightly different from the placental cadagreen, which is important for attachment to the uterus. Like dynamic changes in cadagreen production is very important when the fertilized oocyte comes to the uterine walls. It has to get anchored there, otherwise it is not going to survive, it is going to be lost, and that requires cadagreen interaction, attachment of the embryo to the uterine wall. Then you have retina specific one called R cadagreen, nervous system specific one called N cadagreen, and so on. Okay. And the relative amounts of cadagreen matters. Okay, I will give the punchline right here. The different surface tensions are primarily based on the amount of cadagreen on the cell membrane. More cadagreen, then higher the surface tension, therefore more central location in the embryo. Okay. So, that is what we are going to see from the subsequent experiments here. So, they did a series of experiments, we are going to look at one of them. So, here they increased taking the same kind of cells, okay. So, genotypically identical cells and developmentally also the same fate cells. You express different amounts of a given cadagreen, and then you find the aggregate surface tension linearly increases with that. And does that have a consequence? Yes, that is in the next cell. Next slide. So, here you have these two cells, the green and red cells are identical cells, it is not that they are different in any way, just that the green cells express more cadagreen than the red cells. So, as a result the green one goes inside. Okay. So, they did this experiment with variety of cells with different cadagreens and different amounts, and they convinced themselves that the amount of cadagreen present on the cell's surface matters for where a given cell is going to be present in the embryo. So, now we are going to look at some of the specific cadagreens and their role. So, this is a cartoon of the neural tube, okay, mouse neural tube. So, this is the neural crust. So, the neural fold, this and this are going to come together and form the neural tube. So, notochord below and endoderm. Okay. So, this is epidermis. So, from the ectoderm only these two different kinds of cells are going to form. Okay. So, epidermis and the neural tube cells, they come from the same precursor cells. Now, what we are actually going to focus is how these two separate, like how these cells are going to come together, like this is going to go like this is going to go like this, and then they form the neural tube. While these cells are going to separate away, they are not going to fold into a tube, they are going to form a sheet, and that difference is brought by this starting to express something called N cadagreen, and that cadagreen is not expressed by this. Okay. Both will express E cadagreen because they are still embryonic structures, but this one beginning to express N cadagreen matters, and that is shown in the actual embryos here. So, this cross section of an embryo where E cadagreen, you find that on the epidermal cells. Okay. So, this is the same as this. Okay. So, you have the E cadagreen expression, and see this neural tube part starting from the crest to this, they do not express this, instead they have the N cadagreen there, and that is what allows them to separate from the other ectodermal cells, the meaning the epidermal cells, and form this fold. And when you prevent N cadagreen expression, then there is a heap of cells, they do not separate from the epidermis. Epidermis and neural tubes, although they have become different, one is epidermal cell, another is neural cell, but they do not separate, and for that this differential regulation of the cadagreen genes become critical. This has to express the N cadagreen, like this is critical. So, we are going to see more examples of the same. So, this is a frog gastrolog, okay, early stage, earlier than that mouse cross section we saw. So, here what you are doing is here the expression of the N cadagrin in this half of the embryo, okay. So, this portion is prevented. So, as a result the epidermal and neural cells do not separate and they remain mixed, while here they separate neatly, the epidermis goes outside. So, this is a little bit later, this is neurula stage where the neural tube is forming, and you see the top cells, this sheet of cells, they should actually invaginate, and they do not do it when you block the N cadagrin. This is notochord and this is the neural crust cells. So, these have to go in and they do not go. Okay. So, this is a factin, right, yeah. So, this is neural plate and notochord marker. So, the one of the important step where this cadagrin differential regulation is important is where the cells need to migrate to develop to bring in a new shape. One of them is this neural fold to make the neural tube. So, where cells are invaginating, they are going in, that is one, and then we will see other examples. For example, in Drosophila there is a ventral furrow formation where mesodermal cells migrate inward. So, here it is ectodermal cells that are migrating. So, in these kind of situations these migrations are brought about by cell shape changes which are primarily by differentially regulating cadagrin expression, which is again connected to these proteins, small GTPases and non-muscle myosin and their interaction with actin polymerization. Okay. So, these small GTPase expression and their interaction can be regulated differentially, and that is what eventually leads to this. So, we will see this when we get to cartoons that explain this. So, the primary point here is the morphogenetic movements like invagination in this particular case is primarily brought about by changing the cytoskeletal organization as well as cadagrin expression. And in this process these molecules like Rho GTPases and non-muscle myosin play a very important role. So, essentially they reorganize the cytoskeleton and that in turn changes the cadagrin adhesion. So, these are the ones that are very critical for morphogenetic movements. So, we will see another example and then we will get to a cartoon where this becomes abundantly clear. So, this is a SEM image of Drosophila embryo showing you the ventral furrow. Okay. So, it is at the base of the embryo like on your front belly side. So, those ventral mesodermal cells start to migrate inward and that is how this furrow forms. And this migration is made possible by change in the shape of the cells here. So, this is like if you cut here and look at the cross section. So, you see that some whatever is this protein that is differentially expressed, here you do not see that strong and instead you see here. So, this is like basal where you have the basement membrane, this is the apical. So, this protein present apically is present. So, that gets here localized in this particular place. So, this becomes very clear when we get into a cartoon which is in the next slide. Yeah. So, this gives you a good idea of how these changes happen. So, this is the same cross section. So, this one at an earlier stage than this invagination becoming visible. So, here you have like for example, this non-muscle myosin equivalent in the fly myosin 2 present on the basal surface. Okay. So, this is basal, this is apical where you have basement membrane, that is the basal where the cell is attached, and the other side, the exposed side is the apical. Like in your intestine, inside the intestine part would be the basal and what is facing the lumen would be the apical. So, and these transcription factors twist and snail, they are activated only in the ventral, differential gene expression again, and that eventually leads to this non-muscle myosin migrating to the apical side. okay, and that is now going to constrict here, and when it is constricting it is going to open up here. Like if you take a small cloth purse kind of thing and then you have the string and you pull the string, then that becomes coming closer. So that sort of a thing happening here makes these cells open up and they move like this. So simply cell shape change makes them to take finally this shape of the tissue itself. Okay. So the molecular changes are explained here in the the lower panel. Remember I told you about small GTPases and non-muscle myosin and their interaction with actin matters for this, and that is what you are seeing here. So here you have the rho localized on the basal and you have beta catenin as well there. Remember the beta catenin, right? So do not forget this. Yes, which is critical for attaching cadhegrins to the actin. So this is the early stage distribution. You have some beta catenin here, but rho is primarily here. And with this twist snail expression, they now migrate in the ventral cells only where these two transcription factors are expressed. So the subsequent downstream activation of genes lead to the relocalization of the rho and beta catenin to the apical surface on the ventral cells only, not in the rest of the embryo, and that their interaction with the actin tries to pull the actin cytoskeleton closer, and that in turn affects the outside cadegrin as well, and that changes their interaction such that they start to constrict in the apical side and the basal side opens up internally. So this is the kind of process that occurs here as well in these two situations too. Okay. So this is how cytoskeletal change and cadegrin expression change is crucial for morphogenetic movements, and the small GTPase variety called the rho GTPases play an important role in this reorganization of these molecules, and this is happening within the cell so far, right. The snail twist expression is within those ventral cells, but these changes, these morphogenetic movements can happen even for an external cue, and that is shown by this trachea development, the lungs of Drosophila development shown here. So this branchless, okay, so meaning the mutant does not make branches, so therefore the gene is called branchless. So the wild type will be required for branch formation, okay, so that is how usually genes are named. So the branchless cells, branchless is secreted by the surrounding cells of this a sac like structure which is called the tracheal epithelium, and they make these things to move, or actually one leading cell will start to move towards the cells secreting the branchless, and that branchless is what leads to these reorganization from here to here, and that makes the cells to move and they continue to migrate, and other cells simply through their adhesion, okay, via the right type of cadegrin of the right quantity, make them follow the leaders to cells, and then later these change their shape as well. See here, this tube is forming by the migration of these cells, while the next branches are going to happen by these unicellular structures, like these two cells. See, one nucleus here and this cell is, this whole thing is a single cell. So simply cell shape change makes these branches. Clear? So these are not made, these structures are not made by multiple cells. So a single cell branches like this by reorganizing its actin cytoskeleton and the external this cadegrin adhesion. So this is a view of the actual tracheal system of the Drosophila. So, either through differential gene expression within these cells or through external cues, the cell cytoskeletal and cadaverian reorganization is what leads to migration as well as shape changes. Okay, so this is how some of the morphogenesis happen. Is this clear? So, we saw a set of movement that is primarily of epithelial cells. Okay, so far all that we saw is from a sheet of cells. So, cell migration happens readily in mesenchymal cells as well, but the mesenchymal cell migration is somewhat different. And here you have these 4 steps. First the cell will have to polarize, and then you have a leading edge where you are actually the direction in which the cell is going to migrate, where the cell shape changes as if it is going to make a protrusion there or pseudopod kind of structure, and then that attaches to a surface, that is the adhesion to the extracellular matrix in that direction. Then the other side, the posterior, once polarization you have an anterior and posterior. Here let us say the anterior is the one in the direction of migration, and from the other side you need to get rid of the adhesions. So, these are the 4 steps, and how are these 4 steps brought about? So, it is primarily by reorganizing actin cytoskeleton. So, this shows a polarized arrangement of actin. So, this side you do not have much of it. So, you have actin organized into microfilaments in this. So, this is now polarized, and this is your migrating direction. Okay, let us call this as anterior. And then so this eventually leads to it pushes the plasma membrane such that it takes a shape. Let us say you have here, and when you have actin forming here, this pushes the plasma membrane such that you form that sort of a protrusion. Okay. So, this is because of the way you are organizing the actin microfilaments here. Okay. And now this gets attachment. So, you produce cadaverians and other molecules such that it attaches to the ECM here, and that is what you are seeing in this. So, in this particular case what they have made is one large structure called a lamellopodia, and sometimes it could be a single filament like thing what you call as filopodia. Okay. And in this what you are seeing is the different concentration of the filamentous actin to, remember actin is, what is actin? Actin is a globular protein. When multiple globular actins join together they form filaments. So, they could either be filamental or single molecule soluble form, right. So, this blue is the highest of the filamentous and the red is the lowest. So, they are kind of color coded this. So, here you have intermediate concentration. So, you have filamentous actin more in the migrating side, in the lamellopodia or part of it, and this leads to adhesion to the ECM, and then the 4th step is on the other side, like this portion the attachment is detached by producing proteases that would cleave the attachments, and then this is released. So, that is how the cell migrates. It is kind of a somewhat clumsy migration, but it is good enough for cell migrations to happen in the time scales of development. Okay. So, this is how cells migrate normally. So, these migrations as well as what we saw in this particular context, this rho GTPase and actin, these are what is crucial for morphogenetic movements. So, the main message is these do not really involve variety of molecules. Simply by changing the expression levels of different cadaverians, you can have one cell type as we saw in the epidermis and neural plate cells. By either having 2 kinds of cadaverians and then by changing their levels, so you make this suppress and sorting of cells as well as cell migration, all of that is made possible. And these are crucial in different structure formation at different time points. So, these expressions are dynamically regulated. So, the placental cadagrin expression is exactly required when the embryo comes out and it has to attach. So, in that process, in that particular example, if you take, there are other molecules that are also important for this. So, you have to change the ECM constitution as well. So, all of that put together helps that adhesion. So, the type of cadagrin and their spatial localization and their concentration, all of these are spatially and temporally dynamically regulated. So, I stop here because today I wanted to focus only on the morphogenic movement and the role of cadagrins.